Hello, everyone. My name is JC Wash, and I am going to be presenting the paper called The Alteration of Intrinsic Excitability and Synaptic Transmission in Lumbar Spinal Motor Neurons and Interneurons of Severe Spinal Muscular Atrophy Mice by Jelani Sun and Melissa A. Harrington. This is some research background on spinal muscular atrophy. SMA is a genetic disease affecting the central nervous system, peripheral nervous system, and voluntary muscle movement. It is the leading genetic cause of death in infants. SMA involves the loss of nerve cells called motor neurons in the spinal cord and is classified as a motor neuron disease. Chromosome 5 is caused by a deficiency of motor neuron protein called SMN or survival motor neuron. Because of this deficiency, muscles do not receive signals from nerve cells, therefore, they become smaller because they are not getting stimulated. The research statement in question. In this study, researchers investigated the passive membrane properties, action potentials, long lasting plasticity, and intrinsic excitability, spontaneous and miniature synaptic currents in ventral horn alpha motor neurons and intermotor neurons in spinal cord splices from SMA and control mice. The question is, would spinal motor neural networks be altered in SMA mice compared to controls? Some research methods. The mice were maintained under a 14-hour light and 10-hour dark day with a PMI laboratory rodent diet with some water. SMN1 heterozygotes male and female mice were mated to produce pups for experiments. The pups were 6 to 10 days old when they were used for the experiment. Wild type and heterozygous pups were pooled as controlled, while pups with the homozygous mutations formed the SMA group. In order to record the miniature synaptic currents, tetrodotoxin was applied to block action potential for the recording. Also, to record the miniature synaptic currents, there was a spinal cord slice preparation. With this, a single mouse of 6 to 10 days old was put under in a sealed chamber containing an inhalation isoflurane in an upward flowing biological safety level 2 cabinet. The slices were then moved into the incubation chamber with a transfer pipette and kept at 34 degrees Celsius for 30 minutes. Slices were then left at room temperature for more than 30 minutes before recording. The whole cell patch clamp recording was a chamber that was continuously perfused with preheating 34 degrees Celsius at a rate of 2 to 3 milliliters per minute. The motor neurons and intermotor neurons were identified using an IR 1000 infrared sensitive camera coupled with a DIC equipped BX51 WI microscope. Moving on to the results. For the membrane properties, it was shown that increased spontaneous firing in intermotor neurons and by changes in the membrane properties in motor neurons, the membrane excitability of both motor neurons and intermotor neurons appears to be increased in SMA mice. For the action potential, the results showed that the differences in both passive membrane properties and the action potential suggest an increased excitability of both motor neurons and intermotor neurons in SMA mice. You can kind of see where the action potential was increased in this graph. For the long-lasting plasticity, results show that motor neurons from SMA mice were much more likely to express long-lasting plasticity than controls. No significant differences were observed between SMA and control intermotor neurons. And at the top is the motor neurons, and then at the bottom was the intermotor neurons. So as you can see, there was a difference for motor neurons, but there was not much of a difference for the intermotor neurons. For the miniature synaptic transmission, results showed that the frequency of MEPSCs was significantly increased in motor neurons from SMA mice compared to controls, and there was no difference in frequency of MIPSCs. However, in intermotor neurons, MEPSCs and MIPSCs showed an increase. This increase suggests an increase in inhibitory synaptic inputs onto a subgroup of intermotor neurons that may depress the activity of premotor neurons in SMA. The take home message is that the results showed that spinal motor neural networks are substantially altered in SMA mice compared to controls, and that the differences may contribute to the deficiencies in motor neuron output that has been observed. So the question was answered because the neural networks were altered in SMA mice compared to controls. The synaptic plasticity coexists and functionally interacts with the plasticity of intrinsic properties in most neurological disorders. This is just talking about how plasticity coexists with intrinsic properties. For future research, it says that more research is needed to determine if changes that were observed in the intrinsic and synaptic properties of motor neurons and intermotor neurons contribute the pathophysiology or are part of a compensatory response that helps preserve the function of the motor network. Okay, and that is all. Thank you guys for listening.